Okay, so this little lecture is to remind you of some of the differences and similarities between prokaryotic and eukaryotic cells, and then we will talk about converting units, give you a little math refresher for the millimeter, micrometer, nanometer type units. All right, so main thing to remember is that eukaryotic cells have membrane-bound organelles. Things like the mitochondria and the ER and the Golgi and lysosomes, those all have, you can see the little line, membranes around them or contain membranes. Okay. Compared to a prokaryotic cell, they don't have membrane-bound organelles. So the main one you probably remember is the nucleus, but if you just remember that the nucleus has got a membrane on it, you will remember that eukaryotic cells have those membrane-bound organelles. Prokaryotic cells do not. What's really amazing about prokaryotic cells is they carry out very similar functions to eukaryotic cells. They just do it all in their cytoplasm. So a lot of prokaryotic cells, they actually have inner foldings of the plasma membrane that allow them to do some of the coordinated pathways, things like electron transport chain and cellular respiration, things like that. Okay, but they don't have separate organelles. What I want you to remember, though, is that there are quite a few similarities between eukaryotic cells. So, all similarities between eukaryotic and prokaryotic cells. So, all cells have ribosomes. Ribosomes are essential for protein synthesis. We will get into this later this semester. Okay. All cells have, so there's ribosomes there, cytoplasm, the fluid that allows things to move around in the cell. All cells have DNA. And we'll talk in a minute about how the DNA in eukaryotic cells is different than DNA in prokaryotic cells. And all cells have a plasma membrane. So there are many similarities and differences between prokaryotic and eukaryotic cells. One of the things, I'm sorry, I just think this is cute because my kids used to watch Dora the Explorer when they were growing up. So one thing I want you to note is the difference in DNA. So DNA is DNA, AGCT, but in prokaryotes, the chromosome is circular, whereas in eukaryotes, the chromosome is linear. Why does this matter? Well, if you learned about DNA replication, you learned that there's an issue with linear chromosomes and that we lose the ends of the chromosomes every time the cell is replicating, copying its DNA. And this actually eventually leads to cell death and is a reason that you will someday die. In contrast, prokaryotic cells, DNA is circular. So as they replicate their DNA, it just goes round and around and around, and they actually never lose any genetic information. So a bacterial cell can kind of forever reproduce. Usually eukaryotic cells also have multiple chromosomes. And there are histone proteins that help organize our long strands of linear DNA. In prokaryotes, the DNA is much smaller in the sense that eukaryotes have a lot of extra DNA that we're just now finding functions of, but only about 5% of your eukaryotic DNA, your 3 billion base pairs, only about 5% actually encodes for proteins. Whereas in prokaryotes, a whole lot more encode for proteins. So closer to like 95% of prokaryotic DNA encodes for proteins. So we'll talk later in this semester about protein synthesis and introns. Prokaryotes do not have introns. It's another way they kind of conserve their DNA. So their DNA is really tight. So when I mean smaller, I mean not a lot of extra stuff in their genetic information. Organelles we've talked about, nucleus, no nucleus, membrane bound, not membrane bound, but both have ribosomes, yes, slightly different. We're not going to get into this, but you may have learned that in microbiology if you've taken that class. As I said, our chromosomes in eukaryotes, we usually have multiple, and we usually have them paired, whereas prokaryotes have a single circular DNA chromosome. We will talk about, towards the end of this semester, mitosis. Meiosis you will learn about in genetics. Prokaryotes divide a little differently. So our focus in this cell biology class is really on eukaryotic cells. But I always want you to remember the difference between prokaryotes. And if you're thinking, I forgot what a prokaryote, it's bacteria and archaea. Am I spelling this right? Eukaryotes, plants, animals, fungi, protists, like amoebas and algae. Finally, another difference is that prokaryotes are a lot smaller. Eukaryotic cells in general are a lot larger. Now, there's always exceptions, there's always differences, but we're going to use this range of very small in the micrometer. So this stands for micrometer range. So let's take a look. I love these images because you can do these yourself. And this is just a cheek cell. And you can see there's two cheek cells here. I'm outlining their plasma membrane, and another one over here. And it's big, right? So this cheek cell is eukaryotic. You can see this dark nucleus. And then all these little dots, these dark dots, are bacteria, prokaryotes. So you can see there's quite a size difference between prokaryotic cells and eukaryotic cells. And again, always focusing on this size scale, just to kind of get a feel for what we're looking at. We're going to be down in the micrometer, so our units that we will be using, millimeter, this, or sometimes you see it just written as is micro, and NM, stands for nano. So, most plant animal cells here in the 10 to 100 micrometer size. Most bacteria are in the 1 to 10 micrometer size. Just to get a feel, here's ribosomes, small down to 20 to 30 nanometers. DNA, our double helix, is about 2 nanometers thick and can be long. Right? So you can line up, they say, the DNA in one cell end to end, and it can be like a meter long, and it's all compacted into our nuclei. So again, nanometers, millimeters, and micrometers. And what I want you to understand is the difference between these units are 1,000-fold. So let's look at that. So milli is the largest, then micro, then nano. So if I told you a bacterium was 5 micrometers or micrometers, then you could convert that and say, well, to go to millimeters, we have to divide, to go this way, we divide by 1,000. So it would be 0.005 millimeters. And to go this way, we multiply by 1,000 like it showed. It would be 5,000 nanometers. So all of these represent the same size, 5 micrometers, 0.005 millimeters, 5,000 nanometers. If we were going to write these numbers in scientific notation, 0.005 millimeters is the same as, we go move the decimal 1, 2, 3, 5 times 10 to the minus 3 millimeters. So when you're going this way, it's 10 to the minus 3. If we did 5,000 nanometers, we would say our decimal is, here we go, 1, 2, 3. It's 5 times 10 to the 3 nanometers. So practice with some numbers. Make sure you can convert from millimeters to micrometers to nanometers to millimeters to micrometers to back and forth. As always, let me know if you have questions, but this should help you with some of your Chapter 1 and some of your Chapter 4 objectives. All right, thanks.